Hi viewers, welcome to SEKHAR'S Biofactory for NEET Molecular Basis of Inheritance with Part 5. Please subscribe our channel and click on bell symbol for more information on NEET, KSET and other competitive exams. In this video, I am going to explain the packaging of DNA, central dogma of molecular biology and differences between DNA and RNA. DNA is one of the nucleic acid and the length of the DNA generally it depends on the number of nucleotides or nitrogen bases present in its structure. bDNA is a common type of DNA present in most of the cells, and in this bDNA the distance between two nitrogen bases is equal to 0.34 nanometers or 3.4 angstrom units. Taking some examples, in Escherichia coli, the number of base pairs are 4.6 into 10 to the power of 6 base pairs. In human diploid nucleus, 6.6 into 10 to the power of 9 base pairs are present. 1 nanometer is equal to 10 to the power of 9 meters. Based on this, when we calculate the length of DNA in E. coli, that is equal to 1.36 mm. This DNA it is accommodated in a small area about 1 millimicrons in bacteria cell. Similarly, in human being, when we calculate the length of DNA, that is equal to 2.2 meters. This long DNA is also packed inside the minute nucleus. It is accommodated in a small area about 5 millimicrons in nucleus of human cell. This compact arrangement it is possible because of a packing mechanism or compaction between long DNA along with some proteins. Generally, DNA is acidic in nature. This acidic nature is possible because of large number of phosphate groups present in DNA structure. DNA is negatively charged. It is due to phosphate groups in DNA strand backbone. Coming to package of DNA in prokaryotes, in Escherichia coli, the nuclear membrane is generally absent around the nucleus. In this bacteria cell, the nuclear material it is directly distributed and present in cytoplasm of bacteria cell. This DNA having negative charge, it held with some important special proteins called nucleoid associated proteins NAPs, generally it having positive charge. It forms a structure in bacteria cell, it is called nucleoid. In this nucleoid, DNA is organized in a large loops held by some proteins. Coming to package of DNA in eukaryotes, Nucleosome model. It explains the packaging mechanism in eukaryotes, and it is a scientific model to explain the organization of DNA and associated proteins in chromosomes. And also further it explains that exact mechanism of foldings which are formed in the structure of DNA in nucleus. This nucleosome model was proposed by Roger Korenberg in 1974, and it is the most accepted model for chromatin organization. Coming to nucleosome model, in the structure of nucleosome, some special proteins called histone and non-histone proteins are participated. These histone proteins generally having basic nature. This basic nature is due to two amino acids like lysine and arginine. These lysine and arginine have positive charge. It is due to their side chains. The types of histone proteins participated in nucleosome model. One is H1 protein, number two H2A proteins, H2B proteins, H3 proteins and H4 proteins. These histone proteins, it helps in nucleosome structure. In the structure of nucleosome, H2A protein, H2B protein, H3 proteins and H4 proteins are generally present in pairs. It is called dimer. These dimers are combined to form tetramers, and finally these tetramers are combined to form histone octamer. But H1 protein always present with single, and it is present outside of the nucleosome. These H2A, H2B, H3 and H4 proteins with pair, they becomes into one unit. It is called histone octamer. This histone octamer is also called new body or core. These histone octamers generally carries positive charge, and the negatively charged DNA with 200 base pairs, it wrapped or coiled around histone octamer in two turns. This affinity between histone octamer and DNA molecule, it is possible because of their opposite charges. Along with that, the hydrogen bonds of hydroxyl group in amino acid, phosphate diester bond present in the backbone of DNA molecule, it is important for stabilizing this affinity. The H1 protein generally it is attached over the linker DNA outside of the nucleosome. These units which appears like a small beads, this bead-like structure is called nucleosomes. A small DNA, it connects two adjacent nucleosomes. Now it is called interbead or linker DNA. The nucleosome chain, it gives many beads on string. It appears under electron microscope. DNA packaging at higher level, it requires some additional proteins. These proteins are collectively called non-histone chromosomal proteins or NHC proteins. Based on staining mechanism, the chromatin material is two types. One is euchromatin and heterochromatin. This staining process, it need one stain, that is acetocarmine. This acetocarmine is useful for staining of nucleic acids. After absorbing this acetocarmine, the nucleic acids gets color. In euchromatin, this euchromatin contains a very less number of nucleosomes, and these nucleosomes are loosely packed. With the help of staining, when we provide a acetocarmine to the nucleus, some of the portions they appeared with a light color, it is referred as euchromatin. This euchromatin can easily unwind during replication, and also in transcriptionally it is more active. Coming to second type, heterochromatin. This chromatin contains more number of nucleosomes, and these nucleosomes are densely packed. During staining, it appeared with a dark color and it is densely packed. It cannot easily unwind during replication, and also it takes more time for unwinding. That's why it is transcriptionally inactive. Coming to central dogma of molecular biology. It is proposed by Crick in 1958, According to this central dogma of molecular biology, the unidirectional flow of information from DNA to RNA, RNA to protein is called central dogma of molecular biology. According to the central dogma of molecular biology, DNA have a capacity to produce daughter DNA molecules is called replication. DNA have a capacity to produce RNA is called transcription. This RNA can produce proteins with a process is called translation. But one exemption is there, that is the reverse central dogma of molecular biology. It was proposed by Temin and Baltimore in 1970. This reverse central dogma of molecular biology is also called as Teminism. According to this reverse central dogma of molecular biology, double-stranded DNA, generally it is formed from RNA molecule. It is rarely seen in Rous sarcoma virus RSV. In this, RNA can form DNA. With the help of a process, it is called reverse transcription. In this reverse transcription, one of the enzyme that is called reverse transcriptase enzyme, it catalyzes this reaction, is also called RNA dependent DNA polymerase enzyme. It is the reverse transcription is a main characteristic feature of retroviruses like HIV. And after completion of this reverse transcription, again the double-stranded DNA can transport the genetic information to RNA and proteins. The next topic is the differences between DNA and RNA. Coming to its name, DNA is also called as a deoxyribonucleic acid. RNA is also called ribonucleic acid. Coming to its lifespan, DNA having long lived, but RNA is a very short lived. In their coiling, DNA is spirally twisted to form a regular helix, but RNA strand may get folds at some places to produce secondary helix or pseudo helix like this. Coming to its structure, DNA consists of two strands, but RNA always single stranded, but sometimes it forms secondary double helix structure, but it is only intermittently. The nucleotides. In the structure of DNA, it contains deoxyribonucleotides, but in the structure of RNA, it contains ribonucleotides. The length of the DNA. DNA is a much longer polymer than RNA, but RNA molecule are variable in length but much shorter than long DNA polymer because DNA contains over a millions of nucleotides in its structure, but in RNA, based on its types, it contains nearly 70 to 12,000 nucleotides. The sugars present in DNA is deoxyribosugar. It contains one less hydroxyl group than RNA ribose. But in RNA, it contains ribosugar molecule without the hydroxyl modification of deoxyribose. Coming to base pairs. In the structure of DNA, it contains cytosine, guanine, adenine, and thymine. In the structure of RNA, it contains cytosine, guanine, adenine, and uracil. DNA is generally present inside the nucleus, but sometimes a small amount of DNA is also present in mitochondria. But RNA forms in the nucleolus, then it moves to specialized regions in cytoplasm of the cell depending upon the type of RNA form. It is present inside the nucleus, RNA present in cytoplasm of the cell. Coming to its reactivity nature, due to deoxyribose sugar, which contains one less oxygen containing hydroxyl group, DNA is a more stable molecule than RNA. And also it is useful for molecule which have task to keep genetic information safe. But in the structure of RNA, it contains ribose sugar. It is more reactive than DNA and is not stable in alkaline conditions. RNA larger helix grooves means it is more easily subjected to attack by enzymes. Coming to its function, DNA replicates and stores genetic information. It acts as a blueprint for all genetic information contained within an organism. But RNA, it converts the genetic information contained within DNA to format used to build proteins. Then it moves into ribosomal protein factory. DNA have a capacity to form DNA and RNA, but RNA have a capacity to form only proteins. Coming to hydrolysis, DNA can easily hydrolyzed by DNA's enzyme, but RNA hydrolyzed by RNA's enzyme. Thank you for watching. The next part will be continued in next video.